Hi, and thanks for the invitation to participate. Here I'm going to be discussing some of the emerging methods for studying connectivity and brain dynamics with fMRI. And to start, I wanted to mention that for the last 25 years, correlation analysis has been a fundamental approach for studying brain networks with fMRI. Areas of the brain that show correlated hemodynamic responses are found to align very well with known large-scale functional systems. And recently, we've been able to resolve much finer spatial scale thanks to work by Renzo Huber and others. As we'll discuss more later, it's not straightforward to directly interpret correlated fMRI signals in terms of functional interactions, but overall, correlations or complementary techniques such as ICA have provided valuable insight into functional organization. As a couple of recent directions, in addition to mapping correlation between remote brain areas, there's also been a growing emphasis on the local structure of fMRI correlations, such as boundaries between functional areas, and also recognizing that such boundaries may vary across different people, so we can construct more personalized brain atlases. There are also findings that suggest we may want to represent smoother, more continuous modes of spatial variation, and recent studies have identified gradients that appear to span a functional hierarchy from more primary processing to more abstract cognitive functions. Now, one thing I wanted to highlight is that traditional fMRI analysis has mainly emphasized a spatial perspective. So to analyze the data, typically we take the entire length of our scan, usually several minutes long, and use all of the time points to come up with a single summary map of which regions are most active or most correlated with each other on average over time. However, an emerging direction in fMRI analysis has been to try and delve more into the dynamics of these signals. So fMRI is giving us rich spatiotemporal data. So what if instead of making a single map, we look more explicitly at the patterns that are unfolding over time? And perhaps this may tell us more about interactions between networks or about internal state changes like alertness or attention, and whether features derived from these signal dynamics can provide more useful or more sensitive markers than metrics than we can get from longer time averages. So over the past decade or so, there have been new methods proposed or adopted from other computational fields to try to analyze these dynamics, and this is work from several different groups. So for example, we might take a time window and slide that across our scan and look at what's changing in the covariance between signals or networks. Or we might use a more multi-scale time frequency analysis to see how areas are coupling or decoupling over time. And approaches based on hidden Markov models and related methods have also been used to estimate temporal states in our data. And transient connectivity patterns, such as identified through these techniques, may provide useful markers of brain disorders in which one might expect that dynamic properties of networks are disrupted. And as one example, in recent work with Vicki Morgan at Vanderbilt, we found that in temporal lobe epilepsy, the hippocampus may spend more time connected to specific networks of other regions in patients versus controls. Now, in addition to models based on pairwise relationships between brain regions, another perspective on our data has been to look at more of multivariate snapshots or sequences and whether these exhibit any recurrent patterns over time. For example, here, by clustering activity at different time points, we find that the number and the diversity of patterns that are present in a given scan may reflect cognitive states. And work from Shella Kylehotz's lab has found sequences of propagating activity, including major modes that seem to travel along a cortical hierarchy, such as mentioned earlier, so that brings more dynamic information to that spatial perspective. So overall, there are a growing number of ways for analyzing and representing our data, and this has revealed much more structure than we knew about a decade ago. But at the same time, there are still open challenges to being able to really connect some of our observations to new knowledge of the brain. And to list what I think are a few of those challenges. One is in how we carry out our statistics and our analysis for studying dynamics in fMRI, because we can get spurious dynamics if we're not careful. Another is on unifying different analyses and observations. So to what extent are different methods related to each other and or tapping into similar aspects of the same underlying activity. Interpreting fMRI and dynamic phenomena in terms of functional brain states is another challenge, as well as finding causal circuit interactions. And since this talk is pretty short, I'm going to focus on a couple of examples from these latter areas, but perhaps more can be addressed in the discussion. So one way that we can get closer to interpreting fMRI signal dynamics is to record indicators of a person's physiological or functional state during the scan. And for example, EEG can reveal faster electrical features of brain activity that can indicate levels of alertness and drowsiness. And so this was a study where we looked at short windows of connectivity across a scan, and we found that in periods where the EEG indicated a person was more likely drowsy, there was actually stronger correlation between default mode and dorsal attention networks compared to when subjects were more alert. So one thing that this shows, I think, is that we can start to map transient fMRI dynamics to ongoing brain states that are happening across the course of an individual scan. And another thing this highlights is that the fMRI dynamics themselves may give us a useful window into the internal state of a person. And so in our more recent work, we've been asking to what extent it is possible to detect changes in alertness or other brain states from fMRI itself. Given that there appear to be reliable patterns in fMRI that are linked with alertness, here we developed an approach in which we use spatial pattern information in order to estimate continuous time course of alertness from an fMRI scan. So here is in red is an estimated time course of alertness that's based on the fMRI data alone. And then to validate that measure, we compare it to the actual measured EEG signal in blue. So this is data from one of our subjects. And then further, we wanted to test whether this measure could also capture behavioral variability on a trial-to-trial level. So people can have very variable behavior. Even if you're faced with the same stimulus like a tone over and over, you may have different reaction times. And in part, that may relate to how alert you are. And so what we found was that if you look at in a short time window around each stimulus and if you sort our trials based on whether the subject pressed the button in red or they missed it in blue, we find that our fMRI-derived alertness measure was higher overall in those trials where they responded compared to when they missed. And that was even before the stimulus happened. This is work from Sarah Dale, a PhD student in our lab. And in addition, it seems that that trials if we look at trials where the person did respond, the ones that had faster reaction times were associated with higher pre-stimulus levels of alertness in our fMRI measure compared to trials with slower reaction times. And so overall, the idea is that we may be able to extract vigilance information from fMRI data and at the spatial, or rather at the temporal resolution of the fMRI scan. And then broadly, this has the potential to start adding dynamic vigilance information, even in fMRI data sets where we don't have any EEG or other recorded measure of vigilance, which is the case for a lot of data sets. And then beyond alertness, work from other groups has shown that there are other types of dynamic processes that may be identified in fMRI as well, including affect and memory, spontaneous cognition. And so moving away from a conventional static correlation analyses, I think that a lot of work is showing that we may be able to identify features in our signals that track these dynamic processes. But nonetheless, most of the approaches I just mentioned are based on correlational information in that, how can we start to get more insight into causal network interactions? So there are several potential approaches for that. One is to integrate fMRI with brain stimulation techniques, such as TMS. So this was a study with Amit Etkin's group at Stanford, trying to understand causal influences between major resting state networks. And one hypothesis is that salience and executive control networks may downregulate activity in another network, the default mode network, which is thought to be more internally oriented. And one of the findings was that exciting these salience and executive networks did lead to more of an anti-correlation between these networks and with certain nodes of the default mode network. And of course, there's much more to be understood, and only a few stimulation sites were examined here. But I think that this and other work along these lines also have implications for understanding mechanisms of TMS and finding good stimulation targets in individuals. And then finally, more invasive work in non-human primates can allow for more specific manipulation of neural circuits, and which can be done in conjunction with fMRI. And then perhaps the findings can be translated to human imaging. This is one example. So spatially widespread or global signals in fMRI are often observed in our human imaging studies, but are not fully understood. And here we investigated whether subcortical nuclei that form part of an arousal system, the arousal system, play a role in generating or regulating some of these widespread cortical signals. We looked specifically in the basal forebrain, nucleus basalis, which is implicated in attention, cortical excitability, sleep-wake. And in our experiments, we transiently inactivate this region in one hemisphere, and then we conduct long resting state fMRI scans to see what happens across the brain. And when we did this, we found quite widespread effects. So specifically, we saw that a global component of the fMRI signal was diminished across the hemisphere that was ipsilateral to the inactivated side, and that switched actually when we inactivated the nucleus basalis in the opposite hemisphere. There was also a close connection with vigilance states, which we might have expected since vigilance tends to be associated with an increase in global cortical signals. And so it was, and we found here that it was mainly when the monkey was drowsy that we saw this strong effect of the inactivation. So overall, one conclusion of this work is that disrupting this brain region had substantial effects over distributed fMRI signals, suggesting a potentially key role of this region in generating or somehow modulating global fMRI fluctuations. And if findings translate to humans, then we may gain more information about the circuits that coordinate large-scale hemodynamic fluctuations, as well as how disruption along these pathways, either through natural or pathological processes, may affect fMRI signals and inform biomarker design. So to conclude, fMRI data are very rich, and when we move beyond conventional static correlations, we are able to find even more information in these signals. And here I specifically touched on moving toward more dynamic forms of signal analysis, resolving signatures of ongoing brain states, and steps toward understanding causality through combining imaging and modulation techniques. But as discussed, open challenges still remain. For example, if we start to look at fMRI dynamics, there are many potential features that we can consider, and how can we best identify those which might provide the clearest markers in certain brain disorders. Also, fMRI is a hemodynamic signal, and so how can we best combine that with other non-invasive or invasive technologies to increase the overall information we can obtain about the brain? And as I know we'll hear more about in later talks, a large portion of fMRI signals can be artifacts driven by factors like head motion, and so how to best address those is continuing, is an area of continuing work. And there's also an evolving perspective on what part of that signal is noise and what is an actual valuable signal. So I think that parts of the signal that the field once considered to be noise are now recognized as carrying valuable information. So thank you, and thanks to my lab and colleagues.